right, welcome everybody. And also thank you to those who spoke before because you've helped me with much of this talk. I've got a lot of stuff to cover. Hopefully I'll move at a pace that you can understand and follow, but there's time for questions also afterwards, we hope. All right, so when I think about nutrition in almonds, we think about the big four that matter. Now, we've heard about nitrogen, we've heard about potassium. The other two that fall into the category with these red dots of always managing are zinc and boron. Now, there are other nutrients that on occasions can be limiting in orchards, occasionally manganese, magnesium, a little bit of iron on occasions, but they're not predominant. And those elements, it's local knowledge, you measure, you monitor, and you react accordingly. Zinc and boron fall into the always to be thought about. So asking the very first question, why do nutrient deficiencies occur? And consequently, what do we need to do to fix them and prevent them? And you can think about it at two levels. You can think about first the soil limitations. There are orchards, there are zones where we operate in almonds that have soil conditions that will induce deficiencies. That's a consequence of the pH of the soil, consequence of the chemistry of the soil that alters solubility and mobility of the nutrient. And you can think about things like iron deficiencies, though this is more of a sort of a grape and peach effect than you often see in almonds, but high levels of calcium in the soil, high pH in the soil can limit iron. Zinc, copper to a less extent, deficiencies can also occur in high organic matter soils or very high phosphorus soils. And so we have seen induction of zinc deficiencies with very large levels of organic matter amendments. And it used to be that you would see zinc deficiency at the old corral sites once you convert a feedlot, a grazing property into a almond property. That's kind of historic these days. Phosphorus, calcium, boron deficiencies you get in acid soils. We don't have very many acid soils. Boron deficiency, we'll talk about a little bit later on, you can get in very sandy soils. And we'll talk about how and why that occurs. The other one I'm going to focus more on is what I call about short-term or transient nutrient deficiencies. And there's sort of two ways these can occur in an almond orchard. They can occur when you have very high levels of nutrient demand. So think nitrogen and potassium. If you had 4,000 pounds on the tree, you've got a window of extreme nitrogen and potassium demand that can be difficult to keep up with, particularly if there's simultaneously compromised root growth, which can occur if you've got this high photosynthetic demand for the nuts. What is sort of more interesting, and we'll spend a little bit of time on this, particularly as it relates to zinc and boron, is the demand for these elements that occurs at very specific stages of the biology of the plant. Zinc and boron in particular have an important role in flowering and in bud burst. And clearly that's a very important time in the life cycle of an almond crop. It's when you set the crop. You can learn things from soil pH. Generically speaking, high pH soils limit the availability of elements such as zinc and copper, manganese and iron. And most definitely, if you're up in this greater than 7.5 range, you can think about there potentially being an issue. A few take-home messages, however, are important. You can get iron and zinc and copper soil availability analyses done by testing labs. In general, they're not of much value. Those soluble zinc, soluble iron, DTPA extracted, etc., not a lot of use. What is more use is just generally understanding where you sit in the pH range. For zinc, if you're greater than 7.5, you have a propensity for a greater amount of problems. It doesn't mean you cannot have them when you're lower than 7.5. For zinc and for boron, which we'll show in a moment, really it is leaf analysis, but also your sort of historical experience of do we sometimes see these deficiencies that will tell you whether you're on the margin and you could have a hidden deficiency of importance. In the case of boron, there are a few things that you can get from a soil test. If you've got high levels of extractable boron, Above 1.5, you should start thinking about boron toxicity may be an issue in my future. And you can think of boron a bit like smoking. You know, if 1.5 ppm boron is a cigarette a day, 2 ppm boron is 2 packs a day, 3 ppm is you're smoking the whole carton. It will eventually get you, okay? It's an accumulative toxin over time. You can still definitely get responses to added boron even if you're up above 1 ppm boron. And again, tissue analysis, history, experience is really what's going to tell you, am I on a margin where I can expect a response at least sometimes? So I'm going to focus on this second portion here because it's a little more difficult to understand, and that is that short-term or transient nutrient deficiencies. As I said, you can think about this in particular for nitrogen and potassium when you have very high demand. If you've set a very big crop, if you've got soil conditions that are unfavorable to root exploration. Demand during times when there's restricted growth. So in the cold, wet springs this year, there are a lot of people who ran into a problem with nitrogen because they couldn't get any out on the soil. Cold, wet soils, very little nitrogen turnover, and if you're in the North Valley, you couldn't irrigate or get out until May. That was an example. 2022, we had sort of the opposite problem, hot, dry, smoky season where we had a lot of demand for nitrogen and a difficulty in getting it to the tree. Trees were stressed and not taking it up. We're going to talk about this effect here on zinc and boron in just a moment. All right, so we've seen versions of this slide, and I've shown you versions of these slides over the years. Catherine showed a different version of this. This is the work we did down in Kern County, where we excavated trees throughout the season, estimated when uptake was occurring from the soil. So here we see no uptake from the soil in the first, up until we get mid-March. Dramatic uptake over the next 50, 60 days, and then uptake shuts down after that. This period here is an extreme demand. You could have as much as 250, 300 pounds of potassium or nitrogen being consumed in a 50-day period. There are occasions when supplementation of this with a foliar nutrient or with an additional fertigation application is justified. When you don't keep up with that, you can generate the potassium deficiencies. In this case, mind you, that's a prune tree, but it illustrates that interaction between demand and transient supply, a transient extreme uptake. We're not going to talk a lot more about nitrogen potassium because that's been covered, and I've been asked to cover the zinc. So let's look at transient nutrient deficiencies as they relate most particularly to zinc and boron. Now, I'm not going to talk about these other nutrients, though these can also have this critical period in the growth cycle of the plant where you've got to be certain that you've provided the nutrient of interest. So, an examples here we'll show you in a moment, zinc deficiency, boron deficiency, zinc deficiency here, which in both of these cases for zinc and boron, the critical function is at bud break, vegetative break, floral break, okay? And as you all know, that is really where the most critical phase of the life cycle of the plant exists, of the almond plant exists. If you don't set the fruit, you've got nothing to manage for the remainder of the year. So we'll use this approach for a couple of these slides. I'll show you what some of the deficiency symptoms are, and I'll also focus a little bit on explaining what is the very specific function of these nutrients in plants. And in the case of zinc, the most important and the earliest effect of zinc on the biology of the plant is on RNA, DNA, cell division, and cell growth. And you can understand that if you compromise this, if you compromise the ability of the bud to grow and expand, vegetative, reproductive, you are going to compromise the long-term profitability of that crop. In the case of zinc, you get a clear vegetative system, what's called little leaf, so these little clusters. This is simply a reflection of inadequate zinc to allow DNA, RNA, and protein synthesis to continue. That's why you see these symptomologies and why you see it at this time of year. You see this very clear expression of zinc deficiency. There are a couple of other conditions that can cause this. Thrips on occasion can fool you by looking a bit like this. But this little leaf is a very characteristic zinc deficiency effect. One of the more difficult aspects of zinc deficiency to see is its effect on flowering. And this is some work from Scott Johnson, done in this case in peaches and prunes, not in almonds, but a beautiful picture. This was the zinc deficient plot. This was the zinc adequate plot. And one of the things to think about, here we have the contrast between the plus and the minus, and we can clearly see the effect. If you only had the minus, you would have a field and you'd look at it and you'd say, wow, flowering is kind of weak this year. And you don't have an explicit indication until those leaves come out that this is a zinc deficiency. This is clearly very important. And it leads to the question, if zinc is so critical for this early flowering and leaf out, how do we supply that zinc? How do we get it to where it needs to be? What is the point in the life cycle where the zinc is loaded into those buds? So when during bud development is zinc imported into the buds? When could it be applied to avoid a deficiency? Now in almonds is this phase right here, where you have bud initiation and bud development. So we've done a bunch of experiments recently published here in Plant Physiology, looking at answering that question, when does that zinc get into the bud and when is it remobilized? This work is done down at the Stanford synchrotron, where we use the high electron and high photon impact lines to light up the zinc. So zinc is lit up here. This is red. The red color is zinc. Green color is calcium, blue color is potassium. Going from a young, early developing bud, as soon as that bud starts to develop, you can start to see the accumulation of zinc where next year's flower will be. As you go through a larger accumulation, now you see some calcium accumulating. When you get into a mature bud, very rich in zinc right here at the bud zone, with some calcium and a little bit of phosphorus in the background. So you have a loading up that occurs as those buds start to initiate. In almonds, that may be June, July, August, depending on the cultivar. A reallocation or movement of this zinc into the bud itself as you get towards dormancy, and then a utilization as you go through. So let's look at the utilization pattern. This is a flower going from dormancy through to bud push. This is the external picture. This is a cross section. And we can see here these same colors. So that dormant bud loaded with zinc in the floral tissues in this case. As you move through bud push and bud expansion, you can see that zinc's been remobilized into the flower. Finally, you get a little bit of movement of new zinc into this tissue. And here we have an almost open flower. In dormancy, you can see where the storage is. It's right at that bud apex. A little bit of peripheral storage in the bud axils, a little bit lower down. So all of this really tells you that a flower in the springtime is utilizing that zinc that has been reserved and stored in the previous summertime. Now this has a lot of relevance in almond when you think about it. Bud development occurs during hull split during harvest. If you do a dramatic long dry down, which may be necessary if you don't have a split irrigation system by cultivars, you're undoubtedly compromising this. So this is something we need to think about and not overdoing the dry down. I know why it's done, but at times it gets away from us and it is a compromise. So then how do you put this together and think about managing zinc? You always have to think about the soil, because ultimately the soil and its ability to bind zinc is what causes the deficiencies, okay? And that's what you have to think about. High pH soils, very high organic matter soils, they immobilize zinc. So you have to think about that fundamental cause of your deficiency. Uptake of zinc is very dependent on root exploration and root health. If you compromise root health, you compromise uptake. And some of spring deficiency zinc, when you've got a very cold, wet soil, but the air and the atmosphere warms up and you get a bud burst, you can get a zinc deficiency that's induced by the poor activity of the roots. The key role, as I said, is in that early cell division, protein synthesis. That's why we see the deficiencies where we see them. Zinc is not very mobile in the plant. It doesn't move around except a little bit in the springtime and a little bit in the fall as the leaves start to senesce. What is your fertilization strategy? Thinking about the solubility. If you can manipulate the soil pH, then that is one approach. You've got to, obviously, because zinc is very immobile, got to get the roots and the fertilizers close together. In a moment, I'll show you that there are some net advantages of drip irrigated in a zinc-fixing soil, and that's because of this effect. In terms of specific guidelines, we have a critical value established of 20 ppm, and generally, if your average of your field is less than 30, there are chances that portions of that field are zinc deficient. I'll show you some of that variability in just a second. Soil applications, it's really dependent on soil pH. If you are above 7.5, you will always have trouble with fertilizer applications, and drip irrigation will help, drip fertigation will help. Applications through microirrigation are most effective because they limit this fixation. Frequent soluble applications throughout the season is a good way to go, but as I said, limiting harvest water stress is important, particularly if you are in a zone, in a region where zinc deficiency is prevalent. This is a cause of it. In high pH fixing soils, drip irrigation is better than micro. If you can reduce the soil pH, reduce the soil pH. Foliar applications, in general, they're very inefficient, meaning very little of what you apply gets in. Now, that does not mean they're useless. It means that they should be used as a sort of a solution for a problem you haven't been able to solve otherwise, a supplement to a good drip or micro-irrigation fertigation approach. We did a lot of research, as did Scott Johnson, on foliar fertilizers. They all work as long as they are soluble. You have to have different rates depending on the material, and I know people sometimes choose to use these more expensive forms because they're easier to handle. The old tradition of heavy rates of foliar boron, of zinc, really don't provide any zinc to the tree, okay? If you defoliate the tree, you're going to drop all your zinc on the soil, and that's a really inefficient way to do it. This is an important consideration. This is a field with a 160-acre plot here. You've got a large section of this field that is zinc deficient, okay? If you do a normal soil or tissue sampling where you're only taking a few samples, you could easily miss this. This is not all bad. The sensitivity of this section is sort of like the canary in the coal mine. If you've got some zinc deficiency in a field, you can assume most of the rest of the field is on the margins. That's useful, but this has relevance for how you sample and how you respond. Your zinc deficiency is not across this whole field. The other thing to mention here is we're very concerned about nitrogen use efficiency. If a third of your field is constrained by zinc, then you will be using your nitrogen inefficiently if you provided the same amount of nitrogen across the whole field. Boron. A few things that are different about boron. Unlike zinc, it is uncharged, so it really doesn't get fixed in the soil. Not really any major pH effect unless you get to those real extremes of less than 3 and more than 8.5. This is important. Irrigation water is frequently the most important source of boron. Boron can be leached through the soil, and so we do see most boron deficiency in zones that have good access to good quality water. So boron deficiency is more prevalent on the eastern side of the valley than the west, more prevalent around San Joaquin, Modesto sort of regions. You can get effects on fruit set even if you don't see any vegetative. Its function, just like zinc, is reproduction and vegetative growth. Different than zinc, it's very mobile within the almond plant. This is the only picture I've ever seen of vegetative boron deficiency. You don't see it. Okay, this is an extreme circumstance, 1962. More often, boron deficiency in almonds is just dropping nuts, setting less fruit. You can even get to a circumstance where the tree on the left looks better than the tree on the right because it's not being compromised by having nuts on it and taking away growth. So that's obviously not a good outcome. In terms of boron fertilization, we do have a critical value established in hulls, and I know over the last few years, as I've observed and listened to grower responses, this is a valuable guideline, but it doesn't always tell the whole picture. I have growers who say, I keep putting boron on and it never gets above 100 ppm, but I think I've got plenty of boron. And I've got growers who say, it's always testing 200, but I get a response to boron. So it's a guideline, it's a valuable one. It's not everything. There are some other things going on we don't understand. In terms of soil applications, because boron is not fixed, pretty much any soluble source of boron works, okay? Applications through irrigation water are valuable. Keeping the boron moving, keeping it applied continuously through the growth season, but just like zinc, focusing on applications that are going to deliver the boron to the developing bud is critical. Now, because boron is so easily mobilized into the hull, applications of boron while that hull is growing quickly will often feed the hull but not the bud. Once you reach early hull split, very little new carbon, new boron is going into the hull. Applications then will supply the boron to the bud. So that's a good targeting time. When you talk about foliars, again, boron is soluble and almost all of them work equally well. Boric acid, soluble, whatever it happens to be. You can get boron into buds as they swell. You can't get zinc into a bud as it swells, but you can get boron in, and that's a consequence of it being soluble and uncharged. So we did definitely see benefits of early spring, early flowering applications of boron on seed set, on pollen growth. You've got to be careful. You're not going to get a positive response if you apply boron to an open flower. You get a positive response when you apply boron to an opening flower. Okay? And depending how long your flowering is, depending on when your bees are active, will determine what the ideal application time is. Now, I know that's difficult because soils are wet and you want to co-apply with your fungicides. But co-application with a bloom time spray is not a very effective mechanism of getting boron into flowers, unless, of course, 20, 30, 40% of the flowers haven't yet opened and the bees are going to be active. So there's the dance you have to apply. The other thing we have to be careful about is you've all put boric acid down on the ground to control ants and cockroaches. It's a potent insecticide. Bees are insects. So intuitively, spraying boron onto active bee flight is not likely to be a good thing. So if you think about it all put together, you can think of the phenology of the crop and the times at which you have to be thinking about management. Clearly, getting your nitrogen, potassium, your zinc, and your boron available for that bud push is critical. Likewise, feeding the N and the K during these critical phases is really important. And providing boron, nitrogen, and zinc during bud fill is critical. So think about that interaction. Now, I'm having things waved at me here, and I'm supposed to talk about biostimulants. So file away this foliar nutrient stuff, and we're going to whiz through biostimulants in 2 minutes, okay? All right, so all of you have been bothered by sales guys trying to flog biostimulants, whether they're seaweeds or algae or humic acids or protein extracts or whatever else they are. The question is, do they have a role? Are they just snake oils and things people are trying to sell you, or are they part of this whole move towards a more biological system? You know, biocontrol has worked as an offset to traditional pesticide. Can biostimulants also play a role? So, you know, part of the problem is there's no regulation involved. So I have been working with the the White House Office of Science and Technology and a couple of senators to think about writing a bill. I mean, bill has been submitted. It was submitted in March to regulate these products. And it starts with the definition of these products. So just briefly, a plant biostimulant is a product or a microorganism that acts upon nutrient availability, uptake, or tolerance to stress. And we're going to focus on this guy right here. So, do our orchards undergo stress? And the answer is absolutely. You know, if you think about the genetic and the environmental potential we have, as you go through the season, we lose yield when events occur, okay? And your ultimate yield is never the 5,000, 4,000 pounds that you know this orchard could do sometimes, okay? It's inevitably something less. Where biostimulants are putatively functioning is to mitigate this, to sort of stop the plant from reacting and dropping nuts. And actually this work comes from Sebastian Saar. So, and as an example, you know, heat stress in April can result in June drop. If you could mitigate that heat stress, you'd have less June drop, you'd have more yield. So, do these things work? Now, there are 3 papers available over the last 10 years that did what's called a meta-analysis. That is, they read all of the papers ever written and do an overarching statistical analysis and say, on average, do these things work? Each of these 3 publications come out with a net 18% benefit. Now, that doesn't mean you get 18% benefit every time, but on broad average, that's what's happened. Of course, many times it'll be 0, sometimes it'll be 20. The challenge is how to know under what circumstance and what product you should be using for these for this particular purpose. Here's a trial. There's 150 different trials looking at all the different types of effects that were recorded on this particular product. Some negative effects, a bunch of positive effects, and on average, if you include the price of the material you're applying, you'd say there's a 50-50 chance that you would have no effect. The challenge is identifying what product and what circumstance results in the positive effect, okay? This is the challenge, and when growers ask me, so how do I make a decision, do these things work? Here's a quick summary. Many of them don't, but some do. How's that for useful? Biostimulants are designed to solve stress conditions or enhance nutrient uptake. So therefore, you need to know what stress I'm thinking about. You know, a heat spell in April could lead to June drop. If somebody came with demonstration that I have an anti-June drop high temperature spray, that might be something you try. There's a whole ton of different ones of these, so you cannot say one works, therefore mine is going to work. You got to think about it in the context of your own agronomy. And I'll just reiterate this graph. If you think about where and when challenges exist, you would, of course, first tackle them with your nutrients and your water, but then subsequently think about maybe there's an opportunity for an intervention here. So it's an exciting area, but it's still very new. Thank you.
Thank you, Patrick.